Welcome to another edition of WIP's Biochemistry, and in this lecture we'll be discussing urea cycle. Now, this is actually a continuation of one of the previous lectures on catabolism of amino acids, where we discussed transamination and deamination, by which amino acids release ammonia in all the tissues of the body, and the ammonia will be taken to liver for processing. That is, urea cycle is actually conducted in liver, where the enzymes of urea cycle are present. Now, the ammonia, the detoxification of ammonia. Ammonia is a highly toxic product. It cannot be allowed to circulate in the body. And there are primarily two defense mechanisms involved. The primary defense mechanism is formation of glutamine in peripheral tissues. So, formation of glutamine, and secondary defense is the formation of urea. So, what is the formation of glutamine? So this is what I meant by formation of glutamine. This glutamate formed in peripheral tissues with the help of glutamine synthetase in the presence of ATP. It will be converted to glutamine. So ammonia will be taken up by glutamate to get converted to glutamine, and it travels through bloodstream to reach liver, and it reaches liver and gets converted to ammonium again. With the help of glutaminase enzyme. So this is how ammonia reaches from peripheral tissues, including muscle, to the liver. So which means that the other cells, including muscle cells, leave the burden of processing ammonia to the liver. And urea cycle is second line of defense, and processes the end product of protein catabolism. Mammals are ureotelic, whereas there are animals which are ammonotelic and uricotelic. And this urea cycle was the first metabolic pathway to be elucidated, and that was done by Hans Krebs and Kurt Hansleit. So that's why it is called Krebs-Hansleit cycle. And this is also called, ornithine is one of the substrates involved in urea cycle, so this is called ornithine cycle also. And liver, and to a certain extent, to a lesser extent, kidneys are the organs concerned with the urea cycle. Now, the structure of urea goes like this. It is NH2, C double bond O, NH2. There are two nitrogens and one carbon. One nitrogen actually comes from ammonia itself, which is released in mitochondrial matrix. So that may be released from glutamine or by oxidative deamination in liver itself. So that is one nitrogen, and the other nitrogen comes from aspartic acid that we will see in a moment, as well as the carbon. Carbon is reaching urea from carbon dioxide or bicarbonate. And urea cycle is partly mitochondria. It takes place partly in mitochondria and partly in cytoplasm. And we will see in a moment how much ATP or stoichiometry of urea cycle. So this is actually the overview of mitochondrial process and cytoplasmic process of urea cycle. You can see the ATP involved, bicarbonate involved, ammonia involved in mitochondrion, and that gives rise to carbamoyl phosphate. Then ornithine comes and combines with the carbamoyl phosphate to give citrulline, which goes out from mitochondrion to cytoplasm. So where it combines with aspartate in the presence of ATP, and forms arginosuccinate, and it splits to fumarate and arginine, and arginine actually gets hydrolyzed to give urea, and ornithine formed as going back to mitochondria to complete the cycle. So this is the outline of urea cycle. We will see it in detail in the coming slides. Now, this is a better illustration of the process of urea cycle. You can see the mitochondrion here, and this is the cytosolic part here, and the structures are given. And primarily, in urea cycle, there are 5 enzymes and 5 steps primarily. And the 5 enzymes involved are CPS1, that is carbamoyl phosphate synthetase first, then OTC, this is ornithine transcarbamylase, argininosuccinate synthetase, argininosuccinase, and finally arginase. So these are the 5 enzymes actively involved in urea cycle. And the first step is here, this one here, it actually combines ammonia and carbon dioxide in the presence of the expenditure of 2 molecules of ATP in the presence of CPS first to give carbamoyl phosphate. Let us see the step in detail here. Yeah. Here, one thing which you should remember is that ammonium and ammonia exist in a sort of equilibrium in our body, as well as carbon dioxide and bicarbonate also. So sometimes in some equations this is represented as carbon dioxide CO2. So that doesn't matter here. And this gives rise to carbamoyl phosphate in the presence of CPS first. Now, what happens actually here is that the bicarbonate takes up 1 molecule of ATP. Out of 2 molecules, 1 molecule of ATP is first of all taken up by bicarbonate to give carbonyl phosphate. First of all, the carbonyl phosphate. And then the molecule of ammonia is added to give carbamic acid or carbamate. And 1 more molecule of ATP, the second molecule of ATP, is taken up to give the product carbamoyl phosphate. So that is what happens. It goes through carbonyl phosphate, carbamic acid, and finally carbamoyl phosphate. So now we have got carbamoyl phosphate inside mitochondrial matrix. So like this, carbamoyl phosphate. Now the second step is addition of ornithine, which actually comes from cytosol. So there is an ornithine transport mechanism, co-transport mechanism though. Transporter mechanism is there through which ornithine gets into mitochondrial matrix and combines with carbamoyl phosphate in the presence of ornithine trans carbamylase, like this. So this is the enzyme involved, and what happens here is the from carbamoyl phosphate, the carbamoyl group is transferred to ornithine, to this position of the ornithine, to give rise to citrulline. So you can see here the carbamoyl phosphate, the carbamoyl portion of the carbamoyl phosphate attached to here in the red color. Now we go to the next step, and this is OTC, ornithine trans carbamylase, which catalyzes the reaction second. And so citrulline. So citrulline is formed inside, citrulline is actually formed inside mitochondrial matrix and that goes out to cytoplasm like this. And again, there is another mechanism for transporting or co-transporting citrulline out of mitochondrial matrix into the cytosol. There is a co-transporter involved, and you can see that ornithine and citrulline, they are actually amino acids, but not proteogenic amino acids, because there are no codons for adding these amino acids to protein. So they are non-proteogenic amino acids. So the next reaction involves citrulline and aspartate. Now, aspartate is aspartic acid. It's an amino acid, as you know. And aspartate here actually brings the second nitrogen into play, the second amino group into play. And this is catalyzed by argininosuccinate synthetase and with the expenditure of an ATP molecule there. And this actually is the third ATP molecule used up in the entire urea cycle. So this is the reaction. This is aspartate that brings the second nitrogen in the form of amino group and adds to the carbamoyl group already there in citrulline to give argininosuccinate. Now, the ATP actually here split to AMP and a pyrophosphate instead of ADP and inorganic phosphate. That usually happens. And this pyrophosphate is believed to drive the reaction forward with the hydrolysis. But traditionally, even in the leading reference book, you can see the expenditure of ATP in urea cycle as 3. So, urea cycle actually, the expenditure involves 3 ATP only. So, that is being followed here also. The fourth reaction is the breakdown of argininosuccinate in the presence of argininosuccinase enzyme to give rise to arginine and fumarate. And fumarate leaves the cycle here, and arginine continues with the cycle. Let us see the reaction. Now, this is argininosuccinate, and what happens is that this portion is left as fumarate, and arginine has got both the amino groups in it, both the nitrogen in it. Now here, so this reaction is catalyzed by argininosuccinase, and fumarate has its own story. We will see in a moment. So this is reaction 4, and here arginine, and you can see here in arginine the NH2 C NH2, and actually urea's NH2 C double bond O NH2. So we have to add just an oxygen there, an arginine, and to release urea. So that is being done here with the help of a water molecule. A hydrolysis is done in the presence of, yes, arginase enzyme, like this. So this is hydrolysis, arginase enzyme, and hydrolysis actually releases urea, and oxygen is added from water molecule so as to release urea, NH2, C double bond O, NH2, and what remains is ornithine. And ornithine here completes the cycle, so it's free and can get into mitochondria so as to continue the cycle. So this is what happens, and urea is left in cytosol, so it can through hepatic vein it can reach kidney, through the bloodstream it can reach kidney, and it can be excreted through urine. Now fumarate is here, and fumarate has got its own story. Let us see. And in this figure, you can see the arginine or succinate getting broken down into arginine and fumarate. Arginine, as we know, it goes on to release urea, whereas fumarate, which is left in cytoplasm, it actually gives rise to malate with the help of cytoplasmic malate dehydrogenase. Now, malate can go back to the mitochondrial matrix, or it can give rise to oxalate state, and oxalate state, as we know, it's a substrate for gluconeogenesis. And so in cytoplasm it can give rise to glucose through gluconeogenesis. On the other hand, it can also give rise to aspartate or regenerate aspartate, which can be fed to urea cycle. And this is actually done by the process that we already studied, that is transamination reaction. Now, transamination, which involves the aminotransferase, namely aspartate aminotransferase, AST. So oxalate gives rise to aspartate, which can go back to continue the cycle of urea cycle, urea production. And sometimes this entire process is referred to as urea bicycle also because of this process. And the energy calculation, the whole reaction can be summed up like this. You can see as I said, 3 ATP is being utilized or used up, and aspartate, which brings the second nitrogen, second amino group, is left as fumarate. In fact, the aspartate and the fumarate structures, when we compare, fumarate is the carbon skeleton of aspartate minus the amino group. And there are two H2O molecules utilized. One of them is utilized for the hydrolytic breakdown of arginine, and the other for producing bicarbonate with carbon dioxide. So that's why I said there is an equilibrium, sort of equilibrium between carbon dioxide and carbonic acid, as well as an equilibrium between ammonia and ammonium ion in our body. So this is the energy calculation with the expenditure of 3 ATP. But in true sense, reactions related to or leading to the urea cycle also cause the production of 2 NADH, 2 reduced molecules, namely NADH. And we know that what is the purpose of NADH is, it can generate ATP molecules. So urea cycle releases slightly more energy than it consumes in the real sense. And these NADH are produced in two ways, as you may recollect. One of the NADH molecule is formed by reduction of NAD plus by the enzyme glutamate dehydrogenase in the conversion of glutamate to ammonium and alpha ketoglutarate in mitochondria itself. So that is the glutamate dehydrogenase reaction, in which, as we may remember, both NAD and NADP can be used for this reaction, but the forward reaction utilizes NAD plus. So NADH is formed. Then another one is fumarate, which is released in cytosol, gets converted to malate, and then malate gets converted to oxaloacetate. And this oxaloacetate production is actually catalyzed by another dehydrogenase enzyme. So this dehydrogenase enzyme again releases NADH in cytosol. So totally, when we calculate, totally 4 ATP can be produced with the help of NADH that is released inside mitochondria, as well as the 1 NADH released in cytosol. So that's why we say that the urea cycle is actually slightly more energy. It produces slightly more energy than it consumes. And regulation of urea cycle. It can be coarse regulation or fine regulation. Coarse regulation is always concerned with an enhanced number of amino acids that are needed to be removed. For example, enzyme levels can change with the protein content of the diet. A protein-rich diet can give rise to or release a huge quantity of amino acids to be removed, or during starvation also an enhanced protein breakdown can result in release of more amino acids which needed to be removed from body. Whereas fine regulation is concerned with the enzyme CPS first, carbamoyl phosphate synthetase first, the first enzyme, the rate limiting enzyme of urea cycle, and the N-acetyl glutamate is an allosteric activator, as well as arginine also is an activator of this particular enzyme. Now, N-acetyl glutamate, the activator, is actually synthesized from acetyl coenzyme A, and glutamate in a reaction for which arginine is the activator. Now, both acetyl coenzyme A and glutamate, acetyl coenzyme A actually can be produced from ketogenic amino acids also. So, during fed state, both these acetyl coenzyme A and glutamate are high in availability, so that N-acetyl glutamate can be easily formed, which actually activates urea cycle also. Now, urea is normally excreted through kidneys, and a small amount is lost through sweat also. Now, urea actually diffuses from the liver and is transported in the blood to kidneys, where it is filtered and excreted in the urine. But a portion of the urea that is carried by blood diffuses from blood into the intestine. And in the intestine, it is broken down to carbon dioxide and ammonia, and this is by the action of the bacterial urease present in intestine. So from intestine, this ammonia which is released is partly lost in feces, but significantly a part of it is reabsorbed into the blood. So ammonia now reaches blood. It can reach blood. In patients with kidney failure, plasma urea level will be elevated. So diffusion transfer of urea from blood to the gut, to the intestine, is also elevated. So in such cases, the intestinal reabsorption of ammonia will be high, which lead to hyperammonia cases. The normal blood urea level is 15 to 35 milligrams per deciliter, and this is a very good indicator of renal function also. Like if the value goes beyond normal level, it will indicate renal dysfunction. And urea cycle disorders are there mostly because of deficiency of the enzymes concerned with urea cycle. And this is extremely rare and lead to ammonia intoxication. And there are two cases, the most common, even though this is rare. Hyperammonia type 1, which is caused by the deficiency of carbamoyl phosphate synthetase 1, and hyperammonia type 2 due to the deficiency of ornithine transcarbamylase.